Hello all, I hope you're all doing well and staying safe and healthy. I'd love to be doing this in person, but doing it virtual is better than nothing. So today I'm going to give a quick talk on some of the work that we've been doing in the genus Potamilus. So I titled my talk using molecular approaches to delimit conservation units for multiple species of Potamilus. This is collaborative work that I've been doing with Nate Johnson at USGS, Charles Rancliffe at Texas A&M, and Clint Robertson with Texas Parks and Wildlife. So we all know that freshwater mollusks are some of the most critically imperiled organisms globally. The recent reviews by Hagen Williams in 2014 and Lopez Lima et al. In 2018 did a good job of highlighting North America. So we know that roughly 65% of all the freshwater mussel species are of some sort of conservation concern. Either if you're going by IUCN or if you're going by federal Endangered Species Act. So one thing that seems to be missing as far as how we manage and we recover freshwater mollusks in general, there's not many studies that have gone in and actually tried to delimit population units that are genetically distinctive in order to better manage and recover existing populations. There's been a lot of studies that have shown that there is strong genetic structure in many mussel species, but not many people have gone and tried to delimit these to help managers out. So I'm going to talk about some of the stuff that we've done in Potamilus to answer some of these questions. So the genus Potamilus, 6 of the 10 species are of conservation concern, ranging from endangered under the Endangered Species Act to just state threatened, like Potamilus streckersoni that was just recognized a year or two ago. For this talk, I will be including Leptidea within Potamilus. Our recent study in cladistics kind of ironed out the systematics of the group. So just a little bit about all of these taxa that's important for looking into some of the analyses I ran is that we know that most of the life history characters of all these mussels is very similar. They're all presumed to be host specialists with their Glaucidia only attaching the freshwater drum. And they also have what we're calling miniaturized Glaucidia, either whether it's axe head shaped or if it's further accentuated, such as in Leptidea. Most of the species have high fecundity, and they are presumed to have similar modes of infection, whether it be broadcasting or when they're young by maternal sacrifice. So I'm going to look at 3 different species. So I'm going to show you our results for what we looked at in Potamilus inflatus, and then we did a comparative study within Texas to look at Potamilus amphichaenus and Potamilus streckersoni. So I'm going to start with Potamilus inflatus. So Potamilus inflatus is listed as threatened under Endangered Species Act. There's been a lot of research done into where does this thing still occur. There's been a couple of shells picked up in the Pearl River within the past 20 years or so, 25 years. But it's actually presumed extirpated. So historically, it was thought to occur throughout all the Pontchartrain drainages, the Pearl River and the Mobile Basin. But now we know it's probably extirpated from the Pearl, and it's extirpated from a significant portion of the Pontchartrain drainages as well. And really, extant populations are only occurring in the Amite River below I-12. So it's about 40 kilometers. Kim Brown's and Wes Daniel looked into that in 2014. But one thing Kevin Rowe did in his lab in 1998 is that there was significant evidence that there's interspecific variability between those 2 populations. But the problem is, in our case, when we were looking at this, is that how do we really define what's the genetic makeup of that Pearl River population? If that's the major conservation concern, we have to have some sort of evidence-based way to start driving recovery for that population. So one thing that we came up with, this is work I did with Nathan Johnson, is to kind of take a comparative phylogeographic approach and then try to integrate this whole concept of surrogate species. So we know Potamilus inflatus has been extirpated from probably half of its range, but the congeners, either Potamilus fragilis or Potamilus purpuratus, are pretty common in those drainages. So the idea would be, can we use those as surrogate species to kind of get a general idea of what genetic relationships would look like between the populations of Potamilus inflatus? So since they have similar life histories, we assume they'll have similar phylogenetic relationships. over time. And these things are more common. It's easy to collect them. It's easy to sequence, large numbers of individuals. And that could give us some sort of gauge of what is the genetic makeup of some of these populations that have been extirpated. So what we did is we sequenced mitochondrial and nuclear markers. So kind of what we saw was this clear break for all three of the species. Obviously, with Potamilus inflatus, we were only able to include material from the amite because that's the only place we could find them within the Pascagoula+Pearl+Pontchartrain drainages. But for the other two species, we were able to get representatives throughout their known distribution. And what we saw is that really the phylogenetic relationships or the genetic relationships from these haplotypes networks showed a clear break between the mobile and what we're going to call the PPP based off HAG 2010. And then one question I had, there seems to be, varying levels of genetic divergence. Looking at the haplotype network, there might be a little bit more in inflatus, depending on how we look at it. So I was, we know that like effective population size and other characteristics of populations can affect how fast molecules diverge. So I wanted to see, was this a synchronous split? Did all three of these species split at the same time? So we tested that using this PyMS Bayes developed by Jamie Oakes at Auburn. And kind of what we saw is that, yes, when we run this in star beasts, we do see that there's differing estimates of when these populations split. But generally, we found pretty significant support that this was just one vicariant event that split all of these species, which makes sense. So based on these findings, we can kind of assume that the Pearl River population of Potamilus inflatus is probably going to group really closely with what's in the Amite River. So what we decided to do in this instance was to split these out as management units, and brood stock from the Amite could be used to help augment populations within the Pontchartrain drainages, such as Tangipahoa, I'll get into in a little bit, or in the Pearl River. So I'm going to jump ahead. So I'm going to talk about the heel splitters of Texas. So this is Potamilus amphichaenus and Potamilus streckersoni. So both of these species were, streckersoni was listed as threatened by the state very recently. And amphichaenus is actually a candidate for listing under Endangered Species Act. Both of these species have really been extirpated from significant portions of the range. Potamilus amphichaenus is in the Sabine, Neches, and Trinity. It's been extirpated from all of the river systems in a lot of places, but most notably the lower Sabine and Trinity, it is gone. And then within Potamilus streckersoni, from the work we did in 2019, nearly everything upstream of Waco, the species has been extirpated. So kind of one of the ideas about these populations, we don't really know their relationships. So we set out to investigate this clade of bivalves by looking at both genotype-by-sequencing data and mitochondrial DNA to try to get an idea of what conservation units would look like across their distributions. And then we wanted to see if we could gauge how the species were doing in some sort of empirical way. So we use available survey information to try to evaluate spatiotemporal trends in the distribution for all of the populations of both species. So what did we see? I thought that this was kind of an interesting finding with this study. We sequenced ND1, and this was a lot of the data from the 2019 paper. And we kind of saw the clear split between streckersoni and amphichaenus, and some pretty strong structuring with the Trinity River in yellow coming out molecularly diagnosable using mitochondrial DNA from the Sabine and Neches. And we weren't able to split that out with mitochondrial markers. But when we looked at our GBS data set, which is around 12,000 SNPs, we saw that we could split these things out by drainage. Interesting enough, and I think it's something of note, is that these species were about 2% divergent at mitochondrial markers, but these RAD-seq markers, they had an FST of 0.5, meaning 50% divergent. So when we know that they're species, they're pretty divergent. And then within amphichaenus, for example, the Trinity population was 10% divergent from the Sabine and Neches. So there's certainly a strong break between interspecific and intraspecific distance, even when looking at these rad-seq markers. So nonetheless, we were able to diagnose all of these drainages, but we really didn't see much structuring within these drainages, which was kind of intriguing. As far as the spatiotemporal trends, what we saw is that there was this increasing distributional trend over time, but we saw that it significantly correlated with survey effort. So we know that freshwater mussel collections have definitely skyrocketed in the past 10 to 15 years, and that's certainly the case in Texas, starting with Bob Howell's. There really wasn't much done before that. So this wasn't a completely surprising result, but it kind of highlights the need when we're doing these kind of analyses to understand that historical effort certainly was not, certainly not as high as it is now. So kind of the way I thought about this is that novel populations may be uncovered through more robust survey effort. There's still portions of the drainages that, like in the Brazos River, the upper portions of the Brazos River really haven't been surveyed comprehensively to look for heel splitter there, or some of the smaller tributaries in the Trinity and Niches, for example. So it's just kind of evidence that if we increase survey effort, we may be able to detect more populations. So what did we synthesize from that data set? So using our rad-seq data, really relying on that, we decided to split this out into 4 conservation units, each corresponding to a river drainage. So Potamilus, amphichaenus, and Sabine, Niches, and Trinity, all of which should be managed separately, and Potamilus, streckersoni, and the Brazos. So in Texas, there's been this recent push to have these geographical management units, meaning if there's a dam, upstream and downstream of the dam, we should manage those populations separately. And what we saw from including material upstream and downstream of the dams, there really was no molecular basis for that. It's not surprising. It's only been 100 years. If we think about Potamilus in general, they're using a highly mobile host. They have super high fecundity, which has been shown to retain genetic diversity over time. So this wasn't really a shocking study, but as we look at it from a genetics perspective, managing these things at the drainage level is not an issue. But if we're looking at population status or what's numbers, what's the trends in some of these populations, it's not necessarily a bad idea to break the basin up into those GMUs. But as far as reintroduction strategies and recovery, it's not really going to have any genetic consequence if we use, for example, amphichaenus from the lower Sabine to augment populations of the upper Sabine. So what does all this mean for conservation? So I'm going to focus first on inflatus. Pretty much we should just focus our experts on this PPP province. We know there's only one extant population left. It's about 40 kilometers, as I said, south of I-12. And that population is going to have this serve as a source of broodstock for any sort of reintroduction strategies, either to the Pearl River or the Bogachita, where there's been of the very few historical records that have been found, and in the Pontchartrain, such as the Amite, since it's in such a limited area, the Amite, introducing it to more sites on the Amite or in the Comite may be promising. And then the Tangapahoa is a completely separate river that drains into the Pontchartrain as well, that has one historical record. So one thing that needs to be done is kind of a comprehensive look at the genomics of that population. So we need to see what kind of existing genetic diversity is there. How do we properly maintain that amount of genetic diversity? How do we manage it over time? So developing proper genetic management plan before any of this stuff is implemented would be nice. So the big question around inflatus obviously is it extirpated from the pearl? Is it actually extirpated? So right now I'm working with Nate Johnson. We're going to do some eDNA work to see if we can detect Potamilus inflatus from a bunch of survey work we've done with LDWF. I doubt that will be the case, but we want to make sure that we take every stab at it that we can. So then for Potamilus, amphichaenus, and streckersoni, we have empirical evidence from a genomics perspective that we can manage and recover these populations at a drainage level, just from a genetic perspective. So broodstock selection might be a lot less stringent than we thought, right? So if we're not having to pull stops from upstream and downstream dams, and we can just concentrate on using it as a drainage level, obviously there's a lot more healthy populations that we can pull from. So saying that, translocations definitely could be a beneficial recovery option, knowing, especially in the niches for amphichaenus and then the browsers for streckersoni, where we know there's really good populations in the lower areas of those drainages. So with that, I'd like to acknowledge a bunch of people that have helped me with collections, that have helped me doing this research. Obviously, Baylor University, where I got my PhD, Department of Biology and Crafts are supporting me a lot. Charles Radcliffe at Texas A&M, and Mussels of Texas database, where we got all this survey information from. I have a ton of people that have helped me out with collections. You all know that this research wouldn't be possible without y'all's help. And I'd like to definitely thank the funders. So Fish and Wildlife Service, Jeff Powell at the Daphne office, the Baylor University Vice Provost for Research, who supported this RADSEEK work, and where I'm at now, University of Texas at Austin Single Wire Endowment. And I hope you enjoyed the talk. If you have any questions for me, please feel free to reach out, and hopefully I get to see you sometime soon in person.